In this module, we will talk about the second important feature of stirred fermenter. While talking about the basic part of a stirred fermenter, in that module we have seen that body vessel, agitator or agitation system, and a sparger. These three are the basic component of a fermenter. Except we are not talking about the accessories of any fermenter. We are talking about those parts or those components which are directly attached in a fermenter. So body vessel of the fermenter, we know that as our main body is, so the arms and head and the legs are attached to it. And the same way, a fermenter or a body vessel is the main, and then there is an agitation or agitator system that attached to the body vessel. That is a very basic part of a component of a stirred fermenter. As the name indicates, stirred. The stirring is needed because when we have to run our fermentation process in a deep cultivation, then there is a need of stirring. So you can see here that what is the function of the agitation system. So agitation system provide good mixing. So in case of the good mixing, then there is a increased mass transfer. What is mass transfer? Mass transfer can be talked about in the liquid and the bubble boundary layers. So when we talk about the agitation, that provide the equal contact chance of the medium ingredient to the organism, or we can say that organism has the equal chance to go anywhere in the fermenter vessel, having the chance of utilization of the fermentation component or ingredient or nutrients. By the same way, if there is no stirring, though no proper mixing, then what happens? Then there is a depletion of the nutrient surrounding the organism. So on one side there is a depletion of surrounding, and the same there will be the concentration of the secretions which can cause the toxic environment to that organism. So when we talk about the good mixing that have the effect on the mass transfer, mean that have the good for the transfer of toxic element out of the environment from the effect on that have the positive effect to dilute the toxic effect. On other hand, that have the chance of other nutrient to come closer to the organism. So that on other hand, it also provides the appropriate shear that requires the breaking of the bubbles. So when there is a sparger and the air bubbles has to oozes out in the fermentation media, agitation system split those bubbles into a small bubbles. So as the bubble size decrease, then there will be a more surface area. When there will be a more surface area, then there will be a more chance of the oxygen solubility in the medium that ultimately affect on the metabolic process of the organism. As concerned the agitation system, that consists of an agitator and the baffle. So there are the two component of the agitation system. One is agitator and other is the baffle. So baffles not attached to the agitation system, but that is the part of the agitation system. So what baffles do, I will talk about here, that the baffles are used to break the liquid flow to increase the turbulence and mixing efficiency. So as we have seen in our daily life, when we're making any syrup in our home, either milkshake, etc., just look into the bowl of that. There is an ingrowth. So that ingrowth are basically known as the baffles. Sometimes we put the spoon inside that just to make the more turbulence. So if there will be a no baffle, what happens? There will be a continuous layering of that medium. So the medium which have on a periphery that remain on a periphery, and that medium that is in a center that remain in a center. So baffles actually give the equal chance of the medium to come in contact from the outside to inside and inside to outside. So that is only due to the increase in the turbulence. So in this slide you can see that one part is the baffles and other is the agitator. So as concerned the agitator, that one part is the impeller. So that can be a one impeller, that can be 2, 3, it depends upon the size of the fermenter and the design of the fermenter, that how many impellers are attached to the shaft. This is the shaft. And then there is a motor. And motor, we know that motor has to drive this whole system. Along with this, there is a sensor that senses the speed of the motor, by which we have to control the RPM of the agitation. So link of this agitation system, that is through the seal. As I have told you in our previous module, that seal is very critical because there is a continuous process of agitation. In case of that, seal is very critical. So this system, if the seal is not good, then there is a chance of contamination. In case of good seal, then we have a good and reliable agitation system.